Good morning, class 12th children of biology group. Today again we continue with the chapter 14 of biotechnology and its applications. And in video 5, if you remember, we had studied the major applications of biotechnology regarding our health. We had studied how biotechnology is used for making antibiotics, for making vaccines, for getting vitamins or vitamins in large quantities from microbes. Then we had studied how we get steroids and how steroids work for the good of humans. We also studied about the study of biotechnology in getting insulin of human type by using recombinant DNA technology, etc. Today, let's go on in the details of producing human insulin. Now, insulin is a polypeptide hormone which consists of 51 amino acids arranged into chains. And these two chains, you see here, one is bearing 21, whereas the other chain is bearing 30 amino acids. So you see these two chains, one bears how many, children? 21 amino acids, and the other bears 30 amino acids. These two chains are interconnecting. You see the connections between these two chains? These two chains are interconnected by SS bonds. Do you see SS bonds? Or these bonds are known as disulfide bridges. The disulfide bridges, it interconnects the two chains, one chain bearing 21 amino acids and the other bearing 30 amino acids. Then we see in mammals, including humans, insulin is synthesized as a prohormone or a proinsulin. Now what we see here is a proinsulin, this part, and which contains an extra stretch which is known as C-peptide. Now what does it contain? It contains an extra stretch which is known as C-peptide. Do you see this extra stretch which is attached to the proinsulin? Now this extra stretch is called the C-peptide. The C-peptide is not present in pure insulin and is removed during the maturation of proinsulin into insulin. Once pure insulin is made, then the C-peptide is removed. But in proinsulin, this is an extended part of the chain. On July 5th, 1983, there was an American firm, and this firm was called as Eli Lilly. This firm launched a product called Humulin. What was the product that was launched? This product was launched which was known as Humulin by successful transfer of human insulin gene into bacteria E. coli. If you see here, you see that from the human pancreas cells, the insulin gene was incorporated into E. coli. You see the bacteria chromosome here. From this bacteria chromosome, this is the plasmid. The plasmid was removed, and we used the enzymes here, the restriction endonuclease enzyme, which opened up the bacterial cell, and this plasmid was removed. Now in this plasmid we added this insulin gene which we had isolated from the pancreatic cells, and so this became a new kind of a coded plasmid with the insulin gene attached to it. And then this was again put back, reintroduced back, this plasmid vector into the E. coli bacterial cell. And the E. coli bacterial cell with this new reintroduced plasmid vector began to multiply it, and so we get multiple clones of insulin. That is how so much insulin can be produced for treating humans. And then we see that two DNA sequences corresponding to the chains A and B chains of human insulin were first prepared, and they were introduced into the plasmids PBR322. You had studied in the previous chapter about PBR322. Do you remember this diagram, children, of PBR322? Now this was the vector that was utilized, and in this vector the new insulin gene was introduced in E. coli to produce insulin chains. Now in E. coli, beta galactosidase, there is an enzyme beta galactosidase in E. coli which controls the transcription of these genes. Thus, insulin was the first genetically engineered protein to be used in humans, and now it is not only used here but everywhere in the world. Then we also see that drug designing is a very important part of biotechnology. In drug designing, this is an interesting area of research in which we are making significant contribution in the recent years. First, the knowledge of active site that needs to be attacked by drug is acquired. A site is seen, or a place is seen where the drug needs to be used. So first such a site is seen by the scientist, and then the information is utilized to design a drug according to the site where the drug is to be implemented. The particular drug for that particular site is designed, children, and this drug should not only bind to chosen site. This drug, children, it should not only attach itself to the chosen site but also possess all properties required by the drug. Computers are employed to allow automated site directed drug designing. By itself, the drugs are being designed when computers are employed to do it. Then we see that drug designing is done either by blocking enzyme activity or through blocking hormone receptors or inhibition of nucleic acid synthesis. We see that drugs like trimethoprim, which is an antimicrobial drug, is synthesized. Then we see that Penopenolol is a drug which is widely used for treatment of heart diseases. This is made by drug synthesis. Then we see that cimetidine is a drug for treatment of ulcers, that is designed. Then we see pyrimethamine drug which is used for treatment of malaria. Very recently, children, we are using azithromycin or AZT as a drug for AIDS, which has been developed through these drug synthesis techniques. Now we come to monoclonal antibodies. Yesterday we had studied about what monoclonal antibodies are. The antibodies are protein molecules which are produced by enzyme beta lymphocytes in response to foreign molecules called antigens. Children, we had studied this before in junior classes, that when antigens are introduced into the body, then our body sends soldiers against these antigens to fight them. So we see there is a specific antibody for each specific antigen. Whatever enemy enters a body, according to the enemy, the correct antibody or the correct soldier is sent to fight it. Then we see that antibodies are required for research also. In medicines also sometimes antibodies are utilized. Then we see that antibodies are isolated or they are removed from the serum by sacrificing animals after hyper immunizing them with antigens. What happens is we put lot of antigens into the animals, and when they become hyper immunized or their body brings up antibodies against these antigens, then those antibodies that are raised against the antigens by the animal's body are isolated. From hyper immunized animals, the blood serum is taken, children, and antibodies are isolated from this serum. Recently, you must have seen that in COVID-19 cured patients, the doctors isolated the antibodies made against COVID-19 inside the bodies of the cured patients in their serum. So you must have heard in the news that many people who were cured of COVID-19, they were donating serum. Why they were donating serum? So as to use this serum with antibodies against the COVID-19 virus infected people and to help them fight the virus. Then we see that various antibodies get mixed up in the common blood pool, and the heterologous mixture of antibodies appears in the system. Now, when there are so many antibodies which are mixed up in the blood pool, it is very difficult to separate specific antibodies from the mixture of for a specific use or for a specific antigen. So that is why this difficulty of isolating specific antibody has now been removed by a technique called monoclonal antibodies. Monoclonal antibodies is a technique which has helped to isolate certain antibodies away from other antibodies. What is definition of monoclonal antibodies? Children, it is very important that you learn this definition for your board examination. The monoclonal antibodies or MABs are homogeneous immunological reagents of defined specificity so that these can be utilized for diagnosis and screening with ease and certainty. So we see that certain kind of homogeneous immunological reagents are used specifically for diagnosis and screening with ease and with specificity, with certainty against a certain antigen. Then we see what is Hybridoma technology. It is very easy to understand, children. In this technology, it is used to produce immunity through specialized cells. We see in this an antibody producing lymphocyte cell is taken, and this cell, children, lymphocyte cell is fused with a single myeloma cell or the bone marrow tumor cell, which is capable of multiplying indefinitely just like cancer cells. This myeloma cell or the bone marrow tumor cell, it multiplies indefinitely, and so this cell is fused with the lymphocyte cell first. These fused cells are called hybridoma. So if you are asked what hybridoma is, you can now explain this. These fused cells called hybridoma have the antibody producing capability, and in these they have inherited from the lymphocytes, and have the ability to grow continuously like malignant cancer cells. They can continuously grow, unstoppingly grow. And in recent times, the recombinant DNA technique, which you had seen in the previous chapter, children, the recombinant DNA technique was utilized for the production of antibodies. This technique, children, which you studied in the previous chapter of recombinant DNA formation, is utilized for production of innumerable antibiotics. The genes or parts of genes, they are cloned and they are inserted in cells. The genes or their parts, they are cloned, they are inserted in cells, cultured for production of monoclonal antibodies. So till now we have studied in this video. We will continue further in the next video from here. So till then, children, all the best. Thank you.